We left off speaking about the details of what happens at the synapse in terms of the synaptic transmission, right? The transmission of that signal from the presynaptic neuron to the adjacent postsynaptic neuron. And so we went over the details here. So we'll kind of pick up right here and just go over this one more time for repetition, and then we'll get into a few practice questions. So essentially, we've got our action potential arriving at our axon terminal. We've got these synaptic vesicles that have neurotransmitters stored in them. And when the action potential arrives here, it triggers the opening of voltage-gated calcium channels, which evacuates or dumps this neurotransmitter onto the synaptic cleft, right? That gap in between these two neurons. And this is where we have that synaptic delay. So just to connect that to the delay between the action potential getting here and the response on the adjacent cell, that is the time it takes for the dumping of this neurotransmitter to happen. We then see the neurotransmitter bind onto its respective receptor on the postsynaptic side, and that is what causes the transmission of this energy, right, back to possibly electrical energy if another action potential is initiated on the postsynaptic cell, but basically two types of responses that we're going to speak a lot more about going forward today. We then clear the junction either by reuptaking this molecule and recycling it into a new vesicle. This neurotransmitter can also diffuse away from the junction, or we can degrade the neurotransmitter into smaller parts and they can diffuse away from the junction, right? And the clearing of the junction is important in terms of ceasing the signal, in terms of regulating this transmission, so to speak. All right. Okay, so that's kind of the event, just to go over that one more time. So let's look at a few practice questions here, and then we'll speak about those responses on the postsynaptic cell. All right, let's take a look. Okay, excellent. A lot of response for D. What are the specialized junctions that nerve cells use to communicate with each other called? These are synapses. Okay, so that is correct. Pretty straightforward here. Let's look at another one. Final few seconds. All right. Okay, so a lot of response for B. Which ions enter the axon terminals to directly trigger the release of stored neurotransmitter by exocytosis? So this is calcium. So calcium is correct here. right? It's going to enter via those voltage-gated calcium channels, which trigger exocytosis. I did see quite a few responses for A, so make sure you understand why it's not potassium, all right? And even if we were to open up a potassium channel, potassium's ECDF is not into the cell. So potassium is really, really wrong here, okay? It's going to be calcium. Okay, let's get a final question. Right. All right, final few seconds. Okay, let's see. All right, so a lot of response for A. So the 0.5 to 5 millisecond period when the action potential arrives at the axon terminal to response of the postsynaptic cell, which is called the synaptic delay, is due mostly to which step of the chemical transmission. So while most of you selected A in terms of the combined overall, more people got the question wrong. So I really want to go in. Yeah, more people got the question wrong than right. So I really want to kind of go in and explain why the answer is A. Essentially, we have the triggering of exocytosis, okay? So it would be tempting to think that the delay is due to time for diffusion of this neurotransmitter across the cleft, but that's not the case. It would also be tempting to think that it takes time for the calcium channels to open up. That is not the case, okay? It also would be tempting to think that the neurotransmitter needs time to bind to the receptor. None of these three are the case. So B is wrong, C is wrong, D is wrong. The only explanation that we have for that delay between the action potential arriving at the axon terminal and the response that we see on the postsynaptic cleft is the exocytosis process, right? The synaptic vesicles docking onto the membrane, the fusion of the synaptic vesicle membrane with the neuron membrane, right, with the axon terminal membrane, and the dumping of the neurotransmitter, okay? That is what explains that delay, okay? So just make sure you understand why B, C, and D here are wrong, although they look tempting.
Quick question with that one.
Mm-hmm.
So to kind of like add on to A, isn't it the actual like concentration of the calcium, like not just like the channels themselves, but after the calcium has entered those channels and like it has to hit some kind of like concentration? Do you know what that is? Like if there's a certain amount that it needs in order to have exocytosis happen, or?
It's not necessarily a concentration thing, it's a voltage thing. So voltage-gated calcium channels are going to be triggered to open up at a specific voltage. And when they do, calcium is going to rush in. Calcium has a really strong ECDF, similar to sodium. So when they rush into the axon terminal, they depolarize the entire axon terminal membrane, and that's what triggers that exocytosis.